Hi dear students, today we are going to talk about the sequence as well as the structured databases. Some of the databases which we didn't previously explore is one is a protein data bank. The protein data bank, it usually stores the three-dimensional atomic coordinates for biological molecules such as proteins as well as nucleic acids. Though we think that a protein data bank is only storing proteins, it also contains the three-dimensional structures of nucleic acids. And as a result, you can imply that all the data which is being obtained here is obtained from various techniques such as X-ray crystallography, NMR, as well as molecular computer modeling techniques. So let's just have a look on how does the protein data bank look. I think you can have a look at the protein data bank. This is the website of the protein data bank, and here you have different options. As we can see, you can either deposit protein data, amino acid sequences, as well as the 3D structure of proteins into the PDB thing. You can search. You have the to carry out searching of the proteins. You can visualize the proteins, the 3D structure of various proteins can be visualized. You can analyze. You have an option here called the analyze. What is the analyze option? The analyze option is you can take two different amino acid proteins and you can analyze them. What is the correlation between them? That can be done. And see, you can sequence and the structure alignment can be done. Like if if you have two different amino acid sequences from two different organisms and you need to find out if they are being found to be closely related, you can do a sequence and a structural alignment. It's somewhat similar to BLAST, but the difference is that here you are using a what you are using an amino acid sequence, and if it is so if you're using two sequences, you use a BLAST2 sequence, or if you're using many sequences, you can use different other alignment options. You can check whether the protein symmetry is what is the different symmetries. See the you can This is about hemoglobin, streptavidin, and enovirus. The structure quality, the quality of the information that you're given is shown here. Then, like that, different options are there. Now, let's have a look on the PDB structure or the 3D structure. Okay, now the structure, now the quality, it mainly depends upon the coloration that you have. Uh, let's go right now into, yeah. Now this is a 3D structure. You can visualize in the PDB structure itself. How does the protein look like? All these are protein molecules. Now you can see the helixes which have been found over here, and here also helix can be found. Now the latest entries. This is the C terminus domains of mutant bound with the. It's a protein which has been shown over here. Like that, every protein which you submit here in the PDB database, you will get a 3D structure of that. And what are the difference, what do you say, the different amino acids which have been found in each particular protein can be analyzed here. So the deposit, you can deposit also here. In the deposit option, you can take what are the amino acid sequences, and you can always, the proteins might not be found to be active, be present as such. They might be found ligated to various other ligands as such. So the ligands, that's what about the ligands has been written about here. You can search. It's somewhat similar to that of the what the blast search or the nucleotide search that we did earlier, and you can do a basic search now. If it's a basic search, you can, if I give a name now, for example, alkaline phosphatase, I'm just giving a term here. Now you can have a look. I gave a search of alkaline phosphatase, and you can see the different alkaline phosphatases which have been deposited in the protein database has been available here. This is a paper in which the data has been available by X-ray diffraction, and the organism which is from which it has been isolated is Escherichia coli, and the ligands with which this particular protein is being bound that has been provided here. You can see a 3D view of this particular protein also. Now, coming to the next one, you can see all these are X-ray diffraction based ones. Now, let's see below if we have. Okay, fine. The data, all we have got, the list that we have here is all on X-ray diffraction, which we have got, but here it is from another organism called the Bacillus cereus, and the ligands associated with each and every particular protein has been found to be different in this analysis. Now let's just have a look on to the file. You can, now if I click on to each and every file, I'm clicking onto one particular protein, 5XWK. Let's see what's going to happen. See, this is a view when I searched about that particular alkaline phosphatase. This has been isolated from Sphingomonas and it was being further expressed in Escherichia coli. That is the gene of Sphingomonas, which is coding for this alkaline phosphatase, was taken, and that protein purification and the expression of that particular protein was being done in Escherichia coli here. BL21 was the expression system that you use. And here the enzyme is being classified as a hydrolase enzyme, and you can get how much is the data validated. And if you go down, you can see that it consists of chain A. The sequence length is about 562, and mutations are not being present. The gene name is being provided and an entry name is also being given. Now let's explore this. Now here I am showing the PDB structure, that is a three-dimensional in the PDB database itself. I am just showing you a 3D structure of that particular protein called alkaline phosphatase from Sphingomonas. That's what we are seeing here. And you can see here the polymers, they have been shown as cartoonic molecules, but you can see these are all, these are the main databases. Now you can see the amino acid sequences. See, when I click onto one particular portion, what is that particular amino acid is being shown over here. The next amino acid, the next amino acid. You can see the pink line which is going there. Which amino acid is that is also being shown over there. And uh, at the same time, you just look, the ligands they have told, it has been shown as ball and the stick. This is a ball, okay, the ball and the stick, this is a ligand which has been attached to the particular protein. Now if I go to the next one, if I go to this particular location, you can see each uh, onto the onto the protein which you have here, there are certain various ligands which have been attached together, and there are various bonds also been between them, that has also been depicted in the 3D structure. And more detailed view or the 3D view of it is possible. And let's go into this here. This is the overall view. And if you go towards here, you can see that what are the sequences of this particular protein is also being depicted here. M, L, these are all abbreviations of the different amino acids. So using a PDB database, you can, when you give the amino acid sequence of a particular protein, now here the protein which we are viewing is alkaline phosphatase from Sphingomonas, and it is also in complex with an inorganic phosphate, that's what they have told. The 3D structure has been available over here. The amino acid sequence is available. This is the structure summary which we saw earlier, and then I explored the 3D view. Now let's explore the annotations. See, this is a picture which I got when I clicked the annotations that you can have. You can see that it is alpha beta helix which has been present, and it has a 3-layer alpha beta sandwich model, or that is which domains are being present. That is different domains. It is having an alkaline phosphatase domain, 2 alkaline phosphatase subunits, as well as a gyrase domain has been present over here. And what is the function of each and every domain? Now, alkaline phosphatase is saying it is having a catalytic activity, hydrolase activity, ion binding, cation binding, metal ion binding, zinc ion, transition metal, like that. what are the molecular functions of these particular proteins are being given here. That is what a gene product annotation. That is what is the function of that particular protein is also being depicted in this database. Then what is the cellular component? Where is being found? It is an extracell. It's been found in the extracellular region. If it has been found in the intracellular, that is inside the cell, then you will give it as an intracellular component. And different, that's what about, that is the meaning of what is the annotation. Then you will also have the experiment part. What is, what experiment help us to maintain, get the structure, that would also be explained. They're just showing you how you took this particular protein by X-ray diffraction, the crystallization experiments which they carried out, what conditions they did it, that is all clear. You can explore that. And finally you have the sequence here. Now, if you go to look, this is what I got when I checked the sequence. Now, if there is a helix portion, you can see the helix portion, it has been found by residues 52 to 60. Then, if I go to the next one, again, the helix portion has been made up of residues 88 to 97. If I go to check the beta strand, okay, look, this is the beta strand. I'm checking the beta strand. The beta strand is used made of residues 42 to 48. Then if I want to go further, a metal coordination bond has been formed with the residue 49 over here. So like that, by exploring the PDB structure itself, you can find out. Now, if I go to find out here, the actives, the signal peptide. The signal peptide is using the residues 1 to 9. And if I go to look at the active site, the active site are residues 89. Okay, it's a 89 is an active site residue. Then metal binding residue, it is 89. The different chain like structures, the residues are being given over there. Like that, by taking, when you get an information on a particular protein from the PDB database, you can get its structural summary, its 3D view, what are its functions, and how is the structure being obtained, and what is the sequence of it. You can also see. And if you want to download these files, you can download it from here. You have different options. You have FASTA sequence, the PDB format. Now, if you want to see the FASTA sequence, and each format has a different role. I'll be telling about that, what is the role of each and everything in the coming sections. Now, what is a FASTA sequence, when you have to use a PDB format, that we will be explaining in the coming sessions. So, hope so this is clear to you. What is a PDB database? You get a, it is a storehouse of the 3D structures of nucleic acids as well as proteins which have been obtained. Of course, why are you saying about nucleic acids? Because it is from the nucleic acids that you decipher the what, the protein structures. Sometimes the protein structures will be identified by using various other techniques like X-ray crystallography, as in the example as we mentioned over here. Hope so this is clear to you. Thank you for now, dear students.